Thank you very much. It's a pleasure to be here and speak with everyone, and what an incredible audience. I'm going to focus my talk specifically really on using electronic health records, and you can sort of generalize this across our international cohort, I think, to think about health record data that is in electronic or electronable gettable forms and extend beyond maybe what we think of classically as EHRs in the US. And I'm just going to start with this example of our biobank because it's the one I can give the most density of information on. We started our biobank, which we call BioView, in 2007. And this shows the density of information that we have for those individuals in the biobank, much of it beginning before they enrolled in the biobank. You can see we have data back into the 1980s for some individuals. Each point represents the square root divided by 20 of the number of points that we have. So there's a number of more than we can actually see, but otherwise it would just be solid blue across the different domains. A number of these are actually very expensive to capture, as we all know, such as the radiology information. And so those are the kinds of things that we see as advantageous. And the other thing I'll highlight here is, a lot of times we talk about claims data and ICD and CPT codes and just how much other information there is in the clinical documentation, the radiology lab tests, and those kinds of information, which become very important to understanding what is happening with an individual. And this, we are part of a network called the eMERGE network, which will be another source of many examples. There are some other representatives here of eMERGE as well. And each of the sites in eMERGE has a EHR-linked biobank, and we use the electronic health record data as our primary source for phenotypic information to do studies. And then we've also been working on implementing genomic medicine as well. I'm going to focus more on the genomic study piece of this. I mentioned having clinical documents, and clinical documents are a rich source of information, but they primarily come in hard to understand, free text forms. These can be dictated, they can be typed. Most of ours have been typed of late. We just changed to a different electronic health record system, so we're understanding the differences in formats between our homegrown EHR and a vendor-based EHR, and how that changes how you interpret the electronic health record clinical notes like this. The first step we do is we remove identifiers from this. We use the US HIPAA law to identify a set of 17 categories of information, and we highlight those, remove those, as well as other kinds of things, the so-called 18th category of HIPAA that we try to remove as well, such as wife of the governor, those kinds of things that can be identifying, though not specific. And so from there, we can do things like extract medication information, and we do this using natural language processing. And then we can also do more other types of general purpose natural language processing and techniques to identify things like the sections and the findings they found, whether or not they were positive or negative, or whether it happened in a family member or not. These kinds of general purpose algorithms have certain performances around them. They're not perfect. They are getting better, and it's an area of active research in the computer science domain as well. But the difficulty of accessing clinical text, I would say, has hindered the speed of development of many of these algorithms, and that's something I think we as international cohorts can address as well. And so that becomes part of our research-friendly electronic health record. And then there's certain kinds of exposures that you may want to ascertain, like smoking, and we build custom classifiers for things like that use natural language processing, billing codes, and machine learning approaches. So when you develop an electronic health record, you want to do a phenotype in it. I want to give one example. So an easy one would be high blood pressure. Here are some easy thresholds of 140/90 in two individuals. And individual 1 here has values above those thresholds. Individual 2, all the values are below or at that threshold. But in case of patient 1, they didn't have hypertension but had an ankle fracture, and patient 2 does have very well-controlled hypertension. So what you find when you go to classify by a simple variable that defines the phenotype is, you have to add more information. And in fact, if you look at this receiver operator characteristic curve, the worst performing algorithms, no matter how you look at it, what you apply, essentially is just using blood pressure. And you always get better information by adding other sources of information into the algorithm. And so what we found in basics is that billing codes are helpful, and then putting on other sorts of information, such as that from clinical notes and medications as important markers of who actually has a disease of clinical significance, and laboratory and test results provide a way to triangulate around who really has disease. And then you can use a number of different algorithms, be it Boolean logic or counting rules or regression models or more complicated machine learning approaches to find your true cases. In most cases, the process is an iterative one of proposing algorithm, and the gold standard is always looking at the charts. You can imagine if you had the patient in front of you, that could also be a gold standard. In our case, it's usually reviewing the record and looking at the clinical notes and seeing what the physicians really thought, and going through a process until you refine your algorithm that achieves a sufficient positive predictive value to do your association test. So one of our first examples was looking at hypothyroidism in eMERGE, and in this we executed algorithm across 5 different sites, and then we looked at the genome-wide association study, and we found we found a new signal that was a thyroid transcription factor. Here's another example of another GWAS in eMERGE in a modest number of individuals that identified a sodium channel associated with normal, longer cure restoration in people with normal hearts. And so one of the things that Rory talked about is the importance of following people over longer periods of time. And so when you looked at this, we could look in silico about 20 years from when they had a normal heart and show that each copy of the allele demonstrated increasing risk of atrial fibrillation. We can also do these sorts of phenotypes in electronic health record with pharmacogenetic outcomes. This looks at cytochrome CYP2C19 outcomes predicting clopidogrel adverse events. And so people who are poor metabolizers are more likely to have adverse events, and as many of you probably well know. And so we were able to see essentially about the same effect size in electronic health record as from clinical trial data. A lot has been discussed recently about deep learning techniques and more advanced machine learning. This is one of many such studies that's starting to come out recently, where they use machine learning algorithms that were trained over about 120,000 individuals or images to predict diabetic retinopathy or not. The same sorts of things have been shown for a number of different phenotypes, such as mold classification or mortality in an inpatient admission. And in this case, you can see the dots represent the ophthalmologists in the study compared to the performance here. And you can see, in some cases, you can tune the machine learning algorithm, and you would not necessarily. Let's see, this is not this one. There we go. You probably wouldn't necessarily want to be, this clinician here that is have a sensitivity around less than 40%, but high specificity. Another approach is phenome scanning, a PheWAS approach, and this takes, instead of a given phenotype, you can look at a given genotype and look across many different phenotypes. And in this case, this just shows replication of a number of different phenotypes across a number of different domains of known associations. And what becomes more powerful is when you look across the domain of phenotypes, about 1,600 different phenotypes here on the x-axis, and you can see different HLA regions in this case associating with a number of different autoimmune diseases primarily. And then you can look at a given HLA type and show that one HLA type may be associated with risk and protection for different diseases in the same population. Having electronic health records also lets you do deeper phenotyping, and so in this case, looking at individuals with variants that are thought to be potentially pathogenic and rare in 2,200 individuals, we did an eMERGE, and we had a number of rare variants, and then we sent it to 3 different labs. This is a few years ago, so the state of the art has advanced, but you saw a lot of disagreement in what labs would classify at that point as being pathogenic. The movements around ClinVar and ClinGen is improving this with formal standards. But what we can see here is we're able to go back to the chart and actually see who's had an EKG, what their QT intervals are, if they're on any interacting meds, and potentially allow for the future of callback phenotyping as well. Another way to think about reading out that recently published is looking at what we call a phenotype risk score. So if you take a number of features defined for a given Mendelian disease, for instance, you can take different ontologies and map from a human phenotype ontology to EHR phenotypes. And in that process, we can actually, just like a genetic risk score, calculate a phenotype risk score based on the presence or absence of symptoms and their given weight in the electronic health record, and then repeat this across Mendelian diseases. So if you take an example of cystic fibrosis, you can see that individuals with more features, the CF cases, have higher scores than those CF controls with less features. And the features have weights based on their relative importance or rarity in the EHR. And by doing so, you can think of a phenotype of Mendelian disease as a more continuous trait. So the x-axis here is number of standard deviations from expected score for each individual. And these individuals here boxed are diagnosed with cystic fibrosis. And then there's a number of individuals that are not diagnosed with diseases and features with these other variants that have scores that are in the same realm as these patients that are, have two variants in cystic fibrosis genes. So it gives you a continuous readout and makes you, and gives a way of thinking of a phenotype as more a continuous entity that may explain common disease. I'm going to talk briefly about All of Us, the research program, and some of the data aspects here. When you think about all the different kinds of data we're getting in All of Us, there's an opportunity to take those data, and one of the things we're trying to do is link them all together. So the survey data, the EHR data, and data that could come from smartphones is a real opportunity to link those together and create a resource that articulates those on common data standards. And if you look at all the different kinds of data we're going to have across the initial rounds of All of Us, our model is to take those data, both what's coming from our healthcare provider organizations and what's coming from individuals that are not necessarily associated with a given healthcare provider organization through different means. These EHR data will be coming from hundreds of thousands of different EHR systems that will have to align with common data standards. We can also link those up with health surveys, geospatial data, and other, obviously, the things like the biospecimen data. And we hope to build common standards around this and adapt common data models that will make it easier for people to query, and then have layers upon that with APIs, analyses, and tools. As we think about getting individuals from data from individuals who are not part of healthcare provider organizations or who are part of HPO, but may get data and may see physicians outside, we have the opportunity to do something. We've been advancing a protocol called Sync for Science. And so this is something integrated in 4 of the top EHR vendors currently. And it's a standards-based protocol that shares key information driven by the Meaningful Use Common Clinical Data Set model now, and it's undergoing pilot very soon this year. And so we hope this is a modality for sharing data in a standard way. In All of Us, we hope to move from this traditional model of bringing data to the researchers and them downloading in their own environments to making it accessible in cloud environments that many can access. And I think the real power when you think about this model and having passport models to data access, and then thinking about intercalating the data together under common semantics is this idea of taking all of our different resources. Hopefully we can develop similar semantics around what our consent models look like, how we could share data, and then begin to link our clouds together using common semantics. There's an idea being propagated across different cloud vendors and different resources as we make open standards and GA4GH and other groups to having these silos not be so siloed and talk together as a term that is being used called data biosphere. So, and hopefully that would create a power to do these kinds of resources and studies over many millions of people. So thank you very much for the chance to talk with you.
So hello everyone, and many thanks for the opportunity to speak to you about our experience in the UK Biobank of obtaining phenotype and health outcome data, principally from electronic health records and from digital platforms. So just a very quick resume of the UK Biobank cohort to set what I'm going to say in context. This cohort recruited just over 500,000 men and women from the UK in middle age at the time of their recruitment between 2006 and 2010. These individuals all provided consent for a very wide range of health research by both academic and commercial researchers. And they answered a broad range of questions using a touchscreen questionnaire, had a range of physical measures, and provided biological samples for future assays. And then during the course of the next few years, a range of enhancements in all or large subsets of the participants have been conducted. And those include, for example, the collection of large volumes of data from portable wearable devices. Our first experience of this, as you saw earlier from Rory's talk, was in gathering a very large data set of triaxial accelerometry data. And then we're in the process of starting up a project and indeed have gathered the first sets of data from a target set of 20,000 or more individuals with continuous ECG monitoring using a stick-on device which records the ECG over a period of 2 weeks. Sample assays have now been conducted in all or large subsets of the participants so that genome-wide genotyping, as many of you in this room know, has been completed for the entire cohort, as has a biochemistry panel with results released later this year. But excitingly, exome and whole genome sequencing for large subsets of the cohort are now getting underway, and we're having discussions about proteomics, infectious disease assays, and stool microbiome collection in the entire cohort or very large subsets of it. We're also conducting a multimodal imaging project with MR scanning of the brain, heart, and entire body, and carotid ultrasound, and bone and joint DEXA scanning, for whom 22,000 individuals have been re-recruited so far. So this is re-inviting existing participants, and we anticipate that imaging project will be completed in around 2023. But we're releasing the data as we go, so that already 15,000 imaging data sets have been made available for people to start conducting research, and a lot of methodological research to make these complex data into derived variables that are useful for everybody. We also use the web for collecting data direct from participants remotely, and have had a series of web questionnaires. Importantly, all of these data become incredibly valuable when coupled with comprehensive long-term follow-up for a wide range of health-related outcomes. And the individuals have all provided their explicit consent for this to be done through linkage to a wide range of health-related data sets. And finally, this resource is an open access resource for approved research that's in the public interest, and essentially we don't turn down requests that are for data only. So how does one follow up participants in very large prospective cohorts like this? The aim being to identify a wide range of instant diseases and other health-related outcomes. So traditionally, cohorts have tended to use active methods that require participant re-engagement. Smaller cohorts can use face-to-face reassessment for detailed face-to-face gathering of data. Postal or web-based surveys can be used. These types of methods are, relatively speaking, quite expensive and become more expensive with very large cohorts. And they're also, importantly, prone to incomplete coverage and selective loss to follow-up. And they can only be done at particular points in time, and so they will miss cases emerging between assessments, depending on the nature of those assessments. By contrast, passive methods that use linkages to national health records can follow all the participants without the need for their active re-engagement in any data recollection exercise. That makes them efficient and cost-effective. They do require adequate consent at recruitment to enable this, and they do rely on a universal healthcare system with the availability of relevant data sets. And in that respect, in the UK, we're in a privileged position because of the availability of a universal national healthcare system. Of course, one can only detect cases of disease that are diagnosed in a healthcare setting using such methods. And the other challenge is that the data need to be accurate and sufficiently detailed for research studies. So turning to the two principal methods of follow-up used by the UK Biobank, firstly, with respect to web questionnaires, these have been focused on ascertaining more detailed assessment of exposures, or to obtain information on outcomes that wouldn't be available through linking to health records. And because 2/3 of our participants have an email address and therefore can be rapidly contacted, over the course of a few weeks for each of these sets of questionnaires, we can obtain responses from about 150,000 individuals who will rapidly complete and return to us each questionnaire. So this has been used, for example, to enhance the information on dietary intake through a questionnaire that was designed by Valerie Barrell's group, the Million Women Study, and to collect information on cognitive function, mental health, which is not well recorded in medical records, and gastrointestinal symptoms, with further questionnaires on pain and quality of life planned in the future. These can be very useful, not just for collecting snapshots of information, but also for following change over time. But we do have to be careful here because of the issue of selective attrition. And we've looked at this explicitly with the Whitehall 2 study as an example, looking at the collection of cognitive function using a recall of participants to have measurements of cognitive function. And the biggest predictor of subsequent development of dementia is non-return to participate in a cognitive function assessment. So it is important, if we're going to use these methods, that we understand their coverage and issues of attrition. But the principal method of following the health of the 500,000 participants in Biobank is through linkages to National Health Service records. And we're able to obtain regularly updated information that covers a wide range of diseases from NHS datasets in all 3 countries where our participants were recruited, and largely speaking, where they still are. So, for example, we can link to mortality registers, which exist across the entire country for all participants, providing information on date and cause of death. By early 2016, more than 14,000 of the participants had died, and we know about that and what they died of. There's a very robust and mature and well-used cancer registry system, which goes back to the 1970s in all parts of the UK for obtaining information on cancers, the date, the stage and grade, and gradually the information contained within these registries is being enhanced to contain more detail. And again, it covers all of our participants. So there were more than 79,000 cases of cancer recorded in the UK Biobank participants by late 2015. We can also link using similar methods to hospital admissions with dates, diagnoses, and procedures, giving us information on thousands of cases of many instant diseases. But of course, not all diseases managed in hospital. And so we also have been very keen to link to primary care data for which there has not been any particular national source in any of the countries, but through working with the computer system suppliers, we've been able to link to these data for half of our participants, providing information on a huge range of diseases that are not managed through admission to hospital. But these are messy, real-world data. They're not collected primarily for research, and they're not 100% accurate because of a combination of administrative and clinical errors, and it's important that we understand those. They're mainly structured, coded data sets, coded to international classification of diseases and various other systems. And we've engaged with experts advising in a wide range of disease areas to help us understand these data and maximize their usefulness. In particular, in the initial phases of our work, combining these different linked data sources to create derived disease status indicators so that researchers who use the UK Biobank don't have to go into the messy data but can use disease flags to help them on their way and speed up the process of their research. We've been trying to estimate the accuracy and completeness of these data and consider their limitations and think about potential additional sources of unstructured data that might reveal substantially greater, deeper phenotyping information. So just to give you some quick examples, cancers in the biobank ascertained from the National Cancer Registries. You can see here for 3 of the commonest cancers, the numbers that were observed in the cohort by the time of recruitment, incident numbers emerging by 2015, and then predicted to occur by 2022, with information from the registries on date, stage, and grade of cancer. But we've been working with partners across the UK to look at what we might obtain beyond these structured registry data, exploring the feasibility of retrieving additional information for subtyping from histopathology reports, from digitized histopathology slides, and from tumor specimens. Making good progress, but this is something that needs to be done regionally and can't be done yet at a national scale. We've also been dealing with a wide range of non-cancer conditions, focusing on those that are commonest and creating the greatest public health burden. So here's a few examples showing you the numbers of cases that had already occurred within the hospital records and by self-report by the time of recruitment, and then emerging during the course of follow-up. And here I show also the impact of including information from primary care, where we've been able to model the additional number of disease cases we would identify were we able to link across the entire cohort to primary care data. So you can see in some cases, for example, for diabetes, we just about double the number of cases. Similarly for asthma and dementia, where these are cases that are diagnosed first and most commonly in a primary care setting. What about accuracy and limitations? Here's one quick example for you. So for dementia, we've looked at the positive predictive value of routine healthcare data, both by looking at information from published studies, which shows very wide variation, but in the main, these studies are generating positive predictive values in excess of 80%. And also drilling down into information about subtypes. So for Alzheimer's disease, the positive predictive value in general is rather higher than it is for vascular dementia. And interestingly, when we conducted a sub-study within Biobank to look at this, we found very similar results. So returning positive predictive values of around 80% for dementia overall, reasonably high for Alzheimer's disease, but much lower for vascular dementia. We saw from Josh's talk about the wide range of data there is beyond linked coded data, where the structured data are capturing really only about 10 to 20% of the relevant information and are limited for detailed subtyping of disease. So deeper phenotyping of disease is going to require us to access multiple unstructured data sources, the free text of electronic records, complex electrical signaling data, histopathology slide sets, clinical imaging data. These are hard to get, and once we get hold of them, then to extract value from them, we really need to undertake a massive collaborative effort, developing scalable approaches, using crowdsourcing, natural language processing, machine learning, and artificial intelligence to extract the deep phenotyping information from these sources. And finally, I'd like to acknowledge all the scientists who've contributed to Biobank, but in particular the participants and the generosity of our funders. Thank you.
Thanks very much for the invitation and opportunities to speak at this session. What I'm going to share with you is our experience with China Kadoorie Biobank, which is an academic-led collaborative project between Oxford and Chinese Academy of Medical Sciences. So in terms of study design, very similar to UK Biobank, slightly bigger, slightly older, by a few years. So we recruited just over 500,000 within 4 years, during 2004 and 2008, with extensive data collection by laptop-based questionnaire, a range of physical measurements, most importantly with the collection long-term storage of biological samples. And all the participants are being followed up through record linkage. I'll come back to that. And then we're also doing periodical rate survey, a subset. It would be nice to do all 500,000, but it's not feasible, so we do every 5 years. 5-6% random subset. So this is actually a community-based study. So instead of bringing participants to us, we bring study to them. So for the recruitment, we set up assessment center in local community. And then, when participants come in through invitations, they go through various stations, assess stations, which is carefully planned, organized. And for example, when they come, they have to come bring their national ID number, which would be, get all the personal information to facilitate future follow-up. And then it goes through various stations I'm going to go through. So the whole process is carefully managed, like industrial sort of production line, and to ensure the quality study management. And for example, with a laptop interview, we record all the conversations, so you can facilitate training and the quality control. So overall recruitment rate is about 700 to 800 a day, which is 70, 80 per sites per day. And we managed to maintain that throughout the 4-year period and as well. So this is a huge undertaking, and operate remotely, across 10 different regions, Oxford, China. So everything done is actually electronic. This is actually quite critical to ensure the quality and then smooth operation of study. So over the course, we developed over 90 applications, bespoke IT system, because you can't buy off the shelf from somewhere else. So including data collection, the sample, and this is the interview using laptop-based. This is the system we developed to do outcome adjudication, sample retrieval, shipment, processing, storage as well. So this is actually, make sure, all the data are properly integrated and integrated. So after 10 years, studies in excellent shape. We just got 10-year follow-up. So this is a lot of questionnaire data. You can download questionnaire from website. A lot of measurements, baseline, but it will also start adding a lot of new things like linkage to air pollution, and also start to do some assays, pilot study to inform the future large fundings. We've also done genotype 100,000, but the plan is to genotype all 500,000, potentially whole sequence all 500,000. But I think the data is still growing quite rapidly through assays, but also through linkage to including NASA satellite, we're doing that, and the various assays. In terms of follow-up, we have captured over 1,300 different disease types. This is ICD-10 first 3 digits. If you go for decimal point, that'd be more. 43,000 deaths and only 5,000 lost to follow-up, plus almost 900,000 coded episodes of a hospitalization. But with 100 million chargeable items, we haven't really processed. That's a huge database. It's much bigger than we initially anticipated. So in terms of follow-up, I think just as Cathie said, it's largely through passive follow-up, linkage with the death registry, which is very solid in all the study area, and also to the death registrations, disease incidence for cancer, 4 major diseases, cancer, IHD, stroke, diabetes, with a varying degree of completeness quality. And the most important things is linkage to the national health insurance data. We also do a little bit active follow-up, which is targeted to only a small number. They are not insured, tiny number, and also to find out who has moved out of the study area permanently, try to reduce the number. So national health insurance was introduced 2004 and 2009 with almost universal coverage within a few years. It provides multiple disease diagnosis with ICD-10 codes, plus the procedure code. It's all standardized, but it is managed locally, electronically at city level. They follow a similar protocol. But sometimes it lacks certain details, like cancer histology. They didn't test, but couldn't get the results. So I think we need to improve disease outcome phenotype. So where we are after 10 years, and so this is just give you a flavor of what we captured, almost everything. Some in the few 10,000, like stroke is very common. We have a hemorrhagic stroke, we have 10,000, but 8,000 are incident cases. Nearly 90% are actually confirmed by CT/MRI. This is actually really the best, the data we can ever get. And down to some, snake bite, 400, some earthquake deaths. That's one region. That's actually 2008. So I think it captures everything. In terms of data quality, it's pretty good. If you do some analysis looking to a NEPHO, which is a non-alcoholic liver disease, you got very nice association with the adiposity. Blood pressure, one straight line association, even without adjudication, because it's highly reliably diagnosed. But I think we want to do more, especially to improve the disease phenotyping, and through processing, data cleaning, and individual conducted validation and adjudication. It's very labor intensive, but I think it's worthwhile, not for every single disease, but at least for some major disease of public health importance. This is normally followed a pilot study to assess the need, and before we launch quite big ones. I think in terms of disease, when it's reported quite messy, it's for reimbursement purpose. Sometimes you got 10, 20 different disease items. They only give you one ICD-10 code. So we have to actually develop a tool to split them into different disease and then automatically assign ICD-10 code. Initially it was done manually, but eventually we have to come up with a solution. And then all the items can be downloaded onto the local and a handheld device, and then we can actually conduct a independent validation by collecting the original medical records. For example, the system shows a handheld device. You've got all the information, and then you can go through the medical notes, ensure you got everything captured through the camera as well, and including the various treatment, diagnostic tests. And then the information will be uploaded into the system, and then upload into the server, and the doctor can look at, review, and adjudicate independently. So this is we call iCase. And then doctors, we have an expert team working around the clock across different areas. They can independently adjudicate, including for this example, hemorrhagic stroke, not just hemorrhage, but also lobar, non-lobar, and also we can use TOAST classification, trying to come up with a better adjudication. So this is quite labor intensive, but over the last 3-4 years, we have done over 70,000, including 30,000 stroke, 25,000 IHD, and there's cancer, and then over 3,000 CKD. So it's doable. It's a lot of effort, but I think it's doable. And this has greatly improved the disease subphenotyping. And also this also informed our recent GWAS with some discovery on lobar, non-lobar, small, large vessel stroke, instance ischemic stroke as well. So overall, we capture quite a lot. In terms of analysis, we use this traffic light sort of system to inform the analysis. Some just as reported, some actually go through very extensive processing, cleaning, standardization, and the next stage will be the validation and then adjudication. So for most disease, the reported will be quite good, but we also want to move forward with more work as well. So future work, I think, every 6 months we got linked data, so we need to continue to process the new disease outcome. And we also need to process the 100 million chargeable items. Hopefully they can also inform disease phenotyping. We also plan to extending the outcome adjudication to other disease. There are several of them are lined up. And we also need to develop automated algorithm to do electronic collaboratively. Hopefully we can learn from the experience at Vanderbilt University. We'll also start to pilot collection of dischargeable summary pages, which just become available electronically, and also tumor tissue block collection as well, at least for a subset. So I'm going to stop, and with the final page, which is our website, we also start to open up all the data resources for research from around the world to take advantage of these resources. Thank you very much.
Thank you. So our next speaker is going to be Nancy Pedersen, who's going to talk on the value of biospecimen collection and biobanking. Now she's going to be talking via WebEx, so we'll just check that it's going to work. Nancy, are you there?
You can hear me now?
Yes, we can hear you.
Okay.
Okay, go ahead.
Well, I would like to thank the organizers for inviting me to attend. I apologize for not being able to be there. I promise you would prefer not to be exposed to me right now. What I'm going to do is briefly talk about the value of collecting biospecimens prior to the development of disease. There are a number of challenges in both obtaining and maintaining those sample collections in biobanks and getting it right. There are a number of storage issues about sample depletion and whatnot, but certainly there are a lot of opportunities. Now, the ideal would be that one has biospecimens and exposure information collected prior to the outcomes, and that they're also age-relevant, both biospecimens, exposure information, and pertinent to the outcomes. So this is the example of the way we were thinking with the prospective cohort in Sweden called LifeGene, where, as you can see, there would be very different exposures for the infants versus the adults, perhaps very different outcomes that one is interested in. And certainly one would want to have data collection as well as sample collection over time and have them be relevant. Now, I don't really want to preempt the two speakers after me, but we certainly can be getting quite a bit from these biospecimens, everything from DNA and understanding of the genomics through to proteomics, metabolomics, and whatnot. But in order to have them be really valuable, we do need to have them be collected in a standardized fashion, particularly for some of the omics, such as the proteomics and the metabolomics, even to some extent if we're looking into epigenetic influences. So in LifeGene, for example, we have collected a variety of different blood-based samples as well as urine and aliquoted them such that now with some 30,000 participants, we have over 1.5 million aliquots in the freezer. But the important issue is to make sure that we are standardized and have a cold chain from the test center, from the arm to the freezer within a specific period of time. And here you can see our temperature logs that we have. Definitely, this is all facilitated by having automation. And one of the considerations, of course, is that the automation is indeed costly, but in the long run, certainly worthwhile. Just on the side, we do provide information back to the participants on their own webpage about the results from the clinical chemistries that we take. But it's hard to get it right. And let me just give you the example from transcriptomics. Now, there's a lot of talk about transcriptomics and wanting to make sure that we can look at the RNAs that are involved both in protein synthesis as well as in regulation. But there's a big difference between the mRNA and the microRNA, for example. So the quality of the RNA is important. And if you're interested in mRNA, you have to have samples that are preserved. Either you have to be collected them in PAX tubes, or you have to use something like RNA later, et cetera, which is why you can't get the idea of looking at transcriptomics after the fact on samples that haven't been properly preserved. Not only that, but the protocols matter, the time to the freezer, storage time, seasonal effects, batch effects, and whatnot. Now, there are other challenges in terms of biospecimen collection, and let me share something from my own experience in what brought me to Sweden actually some 40 years ago. We were interested in a longitudinal program of aging in Swedish twins and had both questionnaires and what we call IPTs, or in-person testings. And the idea was to follow them for individual differences in health. We had clinical chemistries, hence we had blood at least collected on the individuals at all occasions. Now, when we first started out, it wasn't really that clear about how to best be processing DNA, but we did know we needed to get the zygosities for these twins, and thus we needed blood groups at the time, we thought. And what did we do? Well, we only saved the red blood cells, hence no leukocytes, no DNA left. What was the next thing that happened? Well, we had a freezer thaw, even though we were collecting both whole blood, saving it as such, as well as serum, there went those samples. As a result, we only have blood samples that are adequately preserved and saved, stored, and whatnot, and been using them for some time from only some of those longitudinal locations. Nevertheless, we do have valuable information from baseline biospecimens, at least that first wave where we did have serum that we could be using, or the lipoprotein panels, clinical chemistries, and longitudinal outcomes. So what you see here are examples of cognitive changes in varying levels of lipoproteins and lipid levels for women on the left and men on the right. Now, there are other considerations, and one of the things we need to think about, we harp on having biospecimens collected prior to the outcome, but cross-sectional effects may very well be different or even greater than longitudinal effects. So we may need to be considering, and I would certainly prefer, that our longitudinal that we collect our specimens longitudinally as well. So here's the example. Here are telomere assessments. And what we actually have is telomere assessments longitudinally on men on the left and women on the right. And the dots are connected, so those are individuals. On the right, what you see are the differences for men and women, solid lines, and dependent on a function of their genetic risk score for telomere length. And what we can see is that these cross-sectional differences or mean differences between groups are certainly much, much greater than the longitudinal changes in the telomere length. So much for telomeres being the important predictor of age that was once touted. Methylation has certainly become a topic of great interest. Talk about the EWASs for age-related CPG sites, et cetera. And here we have two examples of the two of the CPGs that are highly related to age. And indeed, we even have longitudinal information from these two sites. On one hand, you can see we've got those that are being hypermethylated, and the other hand, those are being hypomethylated. So that it would be interesting not only to look at this age-wise, but also as a function of that particular outcome that we're interested in. It would be very valuable to have that methylation data both prior to as well as after the outcome of interest. It also speaks to the fact that cross-sectional data are perhaps not the best basis for calculating the epigenetic clock and its prediction of mortality. Nonetheless, I do want to show you one picture here where we can see very big differences. Here's a pair that is discordant for metformin treatment, and thanks to the longitudinal nature of the data, we see large differences. So back to those considerations, I spoke about getting the sample collection right. We also need to think about maintaining our sample collections and how we're using them, partitioning the samples for various uses. In LifeGene, as I mentioned, we have very many aliquots. And one of the things we need to keep in mind is to try to limit the number of freeze-thaw cycles for some sample types. And let me share with you another one of those embarrassing episodes where we thought we were going to be looking at metabolomics in a case cohort design, and we were looking at a number of different disorders and had our controls based on the cohort, and thought that we had this wonderful result for prostate cancer. It's just that the entire result was spurious because those particular cases had been freezed and thawed multiple times because they were so interesting for a number of other studies. A cohort also needs to take a stance on whether or not they are going to be preparing all of the samples for a particular analysis, or if they're only going to allow cherry picking for rare cases. And this speaks to the previous topic as well. But this needs to be thought about. And then one also needs to be thinking about the future. It's fine to have your baseline collection of data, but are we going to use that all at once, or are we going to save perhaps 10 to 20% of the samples for access after a certain period of follow-up, 10 to 20 years? And certainly there are financial considerations as well. It is difficult to obtain funding for infrastructure, and certainly biobanking and biospecimen collections are an infrastructure. Repeated specimen collection would be ideal, but it is costly. And case collections, on the other hand, from healthcare might be readily available, but they already perhaps have the disorder in question, and they certainly need appropriate controls. This underscores the need for prospective cohorts but of course I'm preaching to the choir. One also needs to remember that there are considerable storage and maintenance costs as well. Finally, one of the things we need to think about is, at least in Europe, and I assume throughout the world, we need to keep in mind the ELSI considerations. In Europe, we have the new, what we call GDPR, which is the General Data Protection Regulations, and there's going to be a lot of issues with regard to how those already collected, already stored collections were processed with regard to the original consent, whether or not it was broad enough, whether there should be reconsent, opt-out, etc., particularly if we're interested in repurposing our samples. But there are opportunities, and I wish so much that I could be in all of the discussions for the rest of this meeting, because if we just look at the cohort list at this meeting, there's enormous potential both within and across countries for whatever kind of purpose we're going to take of looking at those biospecimens. In the Nordic countries, we're well situated with national registries, perhaps not as much of the electronic health records as were mentioned just a few minutes ago, but certainly we have information from hospital, both inpatient and outpatient discharge, basically cradle to grave information, and even genetically informed information. That kind of information, coupled to cohorts, and especially those with biobanking, are going to be a wonderful source of potential for collaborations in general. So actually, I think that opportunities abound. Thank you very much.
Thank you, Nancy. I just want to tell you how well the talk came across from here. So clearly we could do all of this without traveling from our homes. I'm going to introduce the next two talks now so that we can go from one to the next. The first on value of genomic information and then the value of other omic information. But I just wanted to kind of talk about the value of procrastination. And that is, don't do today what you could do better tomorrow. And I think that really is important in thinking about the assays and avoiding the kinds of problems that Nancy was alluding to about pulling samples out, doing assays on them, freeze-thaw. And certainly with respect to UK Biobank, the opportunity that we've had from the funders providing funding to assay all of the cohort has made the data so much more shareable, and I think with respect to genotyping has allowed the power of the funders to push the cost of genotyping down. That has not only helped UK Biobank, but has helped many other cohorts. And I think that could well be true now with not just genotyping, but exome sequencing, and all these other omic assays. So I'll hand over now to MattNelson, who's head of genetics at GSK in Philadelphia, who will talk about genomic information, and then he'll be followed by John Danesh, director of the cardiovascular epidemiology unit in Cambridge, who will be talking about other omic information. So, Matt, thank you.
Thank you. Appreciate the opportunity to be here. It's a great topic and an amazing group of people to be around today. Just to clarify, so in terms of genomic data, I'm going to focus my comments today on genetics in particular. So essentially the heritable portion of our genetic code, and leave the rest of the omics to John Danesh in the subsequent talk. Also have to apologize. There are a couple of rough transitions in the slides because I had a file corruption and had to revert to a slightly older version of this slide set. So hopefully nothing crazy happens. I'm going to take a few minutes at the start and give a brief overview of what is the value proposition for genetics from our vantage point. Why do we care about genetics in pharmaceutical research and in drug discovery in particular? So this is just our perspective on this. Then I think I'll spend the majority of my time talking about genotyping versus sequencing, which I think is the key decision that has to get made when you're thinking about an early stage investment in acquiring genetic data in your biobank. And then we'll talk a little bit about the whole exome versus whole genome, and where does the added value versus cost debate kind of play out. I'll make a few comments about the importance of standards and careful QC, but not go into any detail there, and then just finish with some general thoughts on where we are today in the field of genetics, and where I think that we're going, and the importance that scientific culture plays actually in that journey. So we have a real challenge in drug discovery and drug development, in that it is extremely expensive. We saw earlier the slide, and I'll show it again, of the tremendous decreasing costs per base or per human genome for genome sequencing. We've seen exactly the opposite trend in drug discovery and drug development, that as each year goes on, the cost to develop the next new drug becomes more expensive than the year before. Such that the estimates today are that it costs roughly about $2.6 billion for each new approved drug that is approved. And you can see that that's just between 1990 to 94, comparing that to where we had the latest data in 2004 to 2008. It's almost a 3 times increase in the overall cost. A lot of that has to do with the late stage clinical failure. In phase 2 and phase 3, we see very high rates of failure. And as is shown in the upper right-hand corner, this breaks down in a recent study the reasons for those failures. And both in phase 2 and phase 3, most clinical studies are failing due to lack of efficacy. It isn't that the drug is having a deleterious effect. It often isn't even because the drug is not getting into the tissue that it needs to get into, or engaging the target in the way it needs to engage it, but that the hypothesis of the role that target plays in the onset or in the amelioration of the disease was not correct. We published a paper in 2015 showing that in a retrospective analysis of all of the drug development programs, drugs that have gone into clinical trials, and some small fraction of those that came out the other end as an approved drug, we evaluated what percentage of those that only made it as far as phase 1, or made it as far as phase 2, or perhaps made it all the way through approval, what proportion of those, retrospectively, of those target indication combinations had the underlying support of human genetic evidence. And that's talking about the evidence that exists today, or at least at the time we did the study, for that target indication combination. And we saw this amazing trend coming out of the data. So for those target indications that only made it as far as phase 1 and weren't able to progress further, less than 2% of those could we see any evidence of a genetic support for that target indication combination. And as you progress, those target indications are able to progress further through the development pipeline, all the way through approval, we see the proportion of genetic evidence increasing. That led us to be able to estimate that the presence of genetic evidence when a new drug goes into the clinic doubles our chances of coming out the other end with a successful drug. So this provides genetic information as a way of validating that target mechanism relationship when you're at the very beginning of the drug discovery and drug development odyssey, can be a tremendously valuable way for us to prioritize those projects that we take forward, those new drug discovery activities, the indications that we go after, and has the opportunity to save quite a bit in our through attrition and late-stage clinical failures. So there are a number of applications of genetics in early drug discovery and development. I was just describing the ability to create stronger causal connections between the drug target or the gene that we're interested in, and ultimately the disease that we wish to treat. We can also use genetic information using techniques such as Mendelian randomization, which John Danesh is going to touch on in the next talk, to be able to understand the causal relationship that these intermediate omic biomarkers have with the diseases that we're interested in, that both can help us in identifying the most appropriate mechanism or the most appropriate target for a particular disease, as well as identifying biomarkers that can be used both in patient selection as well as in tracking the efficacy of treatment in very early experimental medicine studies. The genetics can also help us better understand disease subpopulations. What are the different causes of disease? What are the different subtypes of disease that may be amenable to different therapeutic approaches? Can we discover those well before we even start treating humans, and use the genetic information to help guide that, so that when we take these into clinical trials, we're giving them to the patients that have the greatest opportunity of benefiting from that treatment. So where do we gain access to this type of genetic information that's going to help drive this drug discovery and development research? So historically, most of this has come from really the upper left-hand corner of this slide, and that's public databases that primarily have been in the form of published genetic associations. A lot of those curated, for example, in resources like the EBI NHGRI GWAS catalog. Sometimes through collaborations, although those can be very challenging to establish in some circumstances. To some extent, we can leverage data that we're collecting within our clinical trials. So, for example, GSK, we have roughly on an average year 10,000 participants complete later stage phase 2, phase 3, sometimes phase 4 clinical trials. And at least GSK, we routinely collect several biospecimens, including genetic material, and we routinely whole genome genotype those individuals. Currently, we have over 100,000 individuals within GSK clinical trials that have been whole genome genotyped. Where we're shifting is on the right-hand side of this. 2017, I think, will be seen as a watershed moment in the field of genetics overall, with the release and publication of the UK Biobank full 500,000 whole genome genotype data. And you talk to anyone in the pharmaceutical discovery arena, and these data are having an immediate and a very important impact on work that we're doing today, on decisions that we're making today. Moving forward, I think very germane, of course, to the conversations that are going on here. I think other EHR-linked data are really going to drive that next generation of genetics research that's going to help us better understand the role of the genes in disease that will help drive new drug discovery. And ultimately, it really is this kind of cross-biobank research we're discussing here that is going to drive, I think, the most important insights to this. And I'll come back to this at the end of my talk, just briefly. So shifting gears. So to genotype or to sequence. And for those who aren't geneticists here in the audience, the distinction between those two, on the right-hand side, by sequencing, we're essentially capturing nearly every single nucleotide or pair of nucleotides in our human genome for each individual that we capture. And you can differentiate the sequencing in two different types that we'll talk about. Exome sequencing, in which we are only capturing that sequence level variation within the coding portions of our genes, which represents less than 2% of our total genome, versus whole genome sequencing, which is essentially capturing all of the sequenceable DNA in any given cell that we have. So this is essentially attempting to comprehensively capture all variation within each individual. Genotyping, on the other hand, with these types of arrays, this is an Affymetrix, PMRI, the Precision Medicine Initiative research array, and this is just a representative Illumina array. What you do here is you capture signposts. So you measure somewhere between 500,000 and 1 million genetic signposts throughout the genome. These are generally relatively common variants, although they're often enriched with rare variants that are thought to be functional or of value to be capturing. But so you're capturing here, whereas here you'll likely capture on the order of 25 to 35 million variants that differ among individuals within the population. whereas on this side you're capturing 500,000 to 1 million of those. However, that's, well, let's see, okay. So I'll get back to how we try to relate those two in just a moment. So if we have the technology to capture essentially every base in our genome, then why is it even a debate as to whether you genotype or do you sequence? It ultimately comes down to cost. There is a very big cost difference between array-based genotyping and sequencing. So based on some conversations that I've had, and I'll revise this lower estimate here on genome sequencing, since it's perhaps at the aggressive level of where we are today. But those genotyping arrays could be carried out for about $50 per sample, per individual that's genotyped. And that gives you those signposts across the genome. Exome sequencing at scale these days is roughly $300 per complete exome. Genome sequencing, the prices vary depending on who you talk to. I had heard some numbers as low as perhaps $600 by the end of this year. I think probably the costs that are probably more realistic are perhaps up in the $800 range, but it's definitely sub $1,000. We've definitely hit the $1,000 genome at this point. And what implications does this have? Well, it impacts essentially, given an available budget, what kind of sample size can we accumulate? And we've already heard in a couple of talks the value and the importance of achieving scale for these data really to take on the maximum value that they have. So they're just to throw out a few different scenarios for you. So let's say if your goal was to collect genetic data on all 100,000 subjects of your existing biobank, if that was its size, so at $50 a genotyping array, that would cost you about $5 million. On the other hand, exome sequencing would be about 6 times more than that, $30 million, and genome sequencing would be somewhere between $60 and $80 million to be able to collect genetic data on the same number of individuals. To flip that around, probably more realistic in most situations, if you have a fixed budget, let's just say you have $10 million to collect genetic data, you can collect 200,000 genotype arrays, 33,000 exome sequences, or somewhere between 12 and 17,000 whole genome sequences, which obviously puts you in a very different place in terms of what are the potential applications or uses of the data that you're collecting. And just to put that into context, we've talked about power in a couple of previous talks today, but looking at using UK Biobank to estimate the prevalence of these diseases in a cohort like that, I have plotted on here the sample size, the power versus sample size for these, and I'm only going up to 3 million because that, when I made this several months ago, felt like a large number, but after the slide we saw this morning, 84 million, I probably should have gone further out. But here's where UK Biobank is today at 500,000, and for something as common as asthma, where there are 60,000 individuals in UK Biobank with a reported phenotype of asthma, it's tremendously powerful to identify even relatively small effect variants in the genome. But as soon as you start to go to things that are just a little bit less common, things that get down into, a large number of the diseases that we're all interested in are down on that 2%, 1%, 0.5% frequency. And once you start getting into that frequency range, you've got to be up in the 5 to 10 million individuals in order to achieve sufficient power to really be able to draw either if you're doing these genome-wide association studies, to be able to identify, well, what are all the genes that seem to be affecting risk of this disease, or if you're interested in a particular gene and its role in a disease or in a range of diseases, to be able to look across those and have confidence that changes in gene expression for that particular gene is not related to a potential adverse event that may have been hypothesized for that gene, or a potential indication that you're interested in going after for a new drug. So size does matter. Coming back to this idea of genotyping versus sequencing, this slide just summarizes the concept of genotype imputation. So even though with a genotyping array you may only have 500,000 genotypes, we're able to use large and growing numbers of haplotype references that are out there, and to be able to resolve those. And because of the correlated structure within the genome, referred to as linkage disequilibrium. Because of that linkage disequilibrium, we can break the genome down into haplotypes, haplotype groups. And so you can do this with the comprehensive sequence data. You can break it down into haplotypes with the genotyped array data, and you can find the overlaps between them, to put it quite simply, and use the sequence data and the overlapping haplotypes to fill in the missing genetic information. So you can take an array that where you measured 500,000 variants, and you can impute out or extrapolate to over 25 million variants for a typical genome-wide association study that's being done today. So there are, even though we can do this imputation, there are trade-offs in sequencing versus genotyping. And you see those trade-offs most starkly when you want to get down into those variants that are at lower frequency in the population. So what I've plotted on here is in green, we have essentially the coverage of all variation by genome sequencing. In orange, the coverage by exome sequencing. And in blue, the coverage that we have for coding and non-coding variation in UK Biobank with the imputed data that were provided last year. So looking first at genome sequencing, it's near comprehensive. You can capture nearly 100% of the standing variation by going with whole genome sequencing. With exome sequencing, you capture nearly all of the coding variation, not all of it, and I have a slide on that in just a moment. But you capture very, very little of the non-coding variation. So if that's all you've captured, then you only have information about the genes, the coding genes that we know about. And we know from essentially the thousands of genome-wide association studies we've done that most of the variants that are impacting many of the diseases that we care about are located in the non-coding regions of the genome. And with imputation, so UK Biobank was genotyped on an array. We're able to recover for common variants greater than 5%, low frequency 0.5 to 5%, rare 0.05 to 0.5%, and what we're calling ultra rare less than 0.05%. For those first three categories, you're really capturing, especially in the coding regions where they've enriched for some of these low frequency variants in the array, you're getting about 80% of all variation out there that you could then test hypotheses around. But once you get into the ultra rare, those things that occur at very low frequency in the population, your opportunity to be able to impute those it gets very, very low. Now, these curves will shift upwards as larger and larger numbers of reference haplotypes are captured through whole genome sequencing, but it's never going to fully approximate the value of collecting the sequence data altogether. So looking just at the coding regions of the genome, so thinking about arrays versus exomes, for example, or arrays versus whole genomes. Our ability to, for example, if what we want to test is what is the effect of essentially losing the function of a gene on the phenotype of interest. And so if you look at losing the function of the gene, that can happen through lots of variants that occur within the gene. There can be some variants that result in a loss of function that are relatively low frequency or rare, but there are also sometimes some that are actually quite common. And when you get to large enough sample size, you're actually able to get down to those loss of function variants that are essentially private mutations to an individual. And so your power to test for the effect of those loss of functions against your disease of interest is going to be largely governed by what we're calling this cumulative minor allele frequency of these loss of function variants. So it's take all of the loss of function variants in the gene and you put them into a bucket and treat them as essentially a loss of function genotype, and you put that into your analysis. Well, what you might immediately recognize was what we showed here on the left-hand side, is that cumulative minor allele frequency of loss of functions is going to be proportional to some degree to the length of the coding region of the gene, because the longer it is, the more opportunities there are for mutation to arise that knocks out the function of that gene. So that's number one. So you're going to have much more statistical power to test for the effect of loss of functions for very large genes, large coding regions, than with small coding regions. When we did a head-to-head comparison of what the cumulative minor allele frequency that we could capture from the UK Biobank array imputed genotype versus the exome data. And from that, we use we extrapolated from the Exome Aggregation Consortium that Daniel MacArthur, who's here, has spearheaded. We find that, so again here for the very common, for genes that achieve a very common frequency of this loss of function, there's not much differentiated in them. You actually get a slightly larger proportion, obviously, in exomes than whole genomes, or than genotypes. You start to see it differentiate a bit more at the low frequency range, less than 5% frequency, and quite a bit more at the rare range. So there are just more genes that you capture by sequencing where you can test this loss of function hypothesis more powerfully using exome data or whole genome data than you can with genotyping arrays. To cast this in a different way, essentially taking that same analysis of this cumulative minor allele frequency, we ask, well, what is our statistical power? How many genes do we achieve greater than 80% statistical power to test an odds ratio effect of 1.5, 2.5, or 5 if we had 450,000 arrays versus 50,000 exomes, 200,000 exomes, or 450,000 exomes? So for loss of functions, or that result in a relatively modest effect on disease, an odds ratio of 1.5, there's not much differentiation between 500,000 or 450,000 exomes versus 450,000 arrays in terms of the number of genes where you could actually demonstrate that association. But as you get to larger effects, which certainly exist out there, you see that the exomes and the genotyping arrays reach parity somewhere around a 2-to-1 ratio. So you achieve the same power with 2 times as many arrays as you do for one exome. So food for thought. The argument in favor of sequencing versus genotyping varies depending on the kind of cohort, the kind of biobank that you're working with. If you, I think that you'd have to think long and hard if you're trying to argue the value of whole genome sequencing today at the costs that are out there of a standard population-based cohort in a large random mating population. But there's some populations for which you get a lot more bang for your buck. For example, consanguineous cohorts, and this is taken from Catheriesen's paper that was published last year on the PROMISE the Pakistani population. There's a similar effort in East London, Genes and Health, that probably shows a very similar kind of curve, that you essentially will accumulate these rare knockouts more quickly in these consanguineous populations. I think I've covered essentially everything that's in that slide. In the interest of time, just want to highlight that standards for how you collect and store and share the data are important. These are real-world examples from my colleague Robert Scott, as he's tried to carry out meta-analyses and just trying to reference the allele frequencies getting from his collaborators versus just the HapMap allele frequencies, and this is what the pattern you want to see, and he's seen patterns like this, patterns like this, patterns like this. None of those are good. Good data curation standards are critical. I'm just going to finish on this comment that it's shocking to think how far we've come in just the last decade in terms of our knowledge about how genes influence all of the traits that we care about, from disease to the more mundane. We have learned so much, and this is just going back. This is kind of the pre-UK Biobank release knowledge here, which I haven't updated this on, which is going to actually be quite a bit more. A lot of this has been driven by the technology, shown here in black, mostly. Our ability to measure more cheaply. But, some of the biggest changes have actually been not the technology, but it's actually been the scientific culture, the culture of being willing to share data, to work together, to agree on common standards, and to bring these things together. I think UK Biobank is a beautiful example of a shifting culture of open and sharing. And just to think about where might we be 10 years from now, if we look ahead, I think that we probably haven't even seen the inflection point in our understanding of how genes influence and our genetics influences traits that are going to be important to us as we tackle the next generation of health challenges. where mostly in the past we've been dealing with case control type cohorts and anthropometrics. I think in the future it's going to be more about understanding disease subtypes and understanding genetic impacts on trajectories of disease, modifiers of disease, and how those can then be leveraged for in clinical practice and in drug discovery and development. And I think a huge part of our being able to do that is, again, the continued development and progression of the maturation of the scientific culture that creates an even more open environment for sharing of data, sharing of information that enables this kind of research. Thanks.
So, good afternoon. Could I have a slideshow on, please? Thank you very much. So, my talk is going to be about cautious optimism. There are 3 very good reports that have come out recently on this topic from the American Heart Association, the U.S. National Cancer Institute, and from the Institute of Medicine. I'm going to have an epidemiological take on this topic, so you might hear more of the caution than the optimism, but there is optimism in the talk as well. So, of course, we use the word omics as shorthand to refer to collectivities. Sidney Brenner once said, perhaps in allusion to what MattNelson just told us, that the only omics that matters is economics. And I think we've just heard why that's so. The collectivity of genetic sequence variation, as you've just heard from Matt, genomics. You've heard earlier this morning from Josh, Cathie, and Zhengming about using electronic health records supplemented with other information for an endeavor that sometimes is called clinical phenomics. You heard a bit from Jeff Ginsberg about monitoring and other devices. And perhaps David Hunter and Walter Willett will elaborate on lifestyle and broader environmental exposures, so-called exposomics. And what I'm going to try to do is to talk a little bit about these molecular traits that some refer to as the express genome or molecular omics, and some examples are shown here. Part of the interest in this is related to biology. So the ability to overlay these different domains of biology, particularly together with genomics, can open new avenues of biological understanding. Part of it is to try to address the post-GWAS grand challenge. We now have some 2,000 or so loci that are associated with disease. But for the large majority, we don't know what the effector transcripts are, what the genes are. And the idea is that overlaying some or all of this information might help localize those genes to give us more rapidly actionable insights. And of course, part of it may relate to how the broader environment mediates these effects. And others, particularly in the space of risk prediction and clinical biomarkers, are more interested not so much in the etiological relevance or the biological relevance, but how well these markers or subsets of these markers can actually predict disease. So if you like, these are the broad areas of interest, or some of the areas of interest that illustrates or suggests the potential value of these approaches. I would suggest that there is reasonably compelling evidence that the first 2 are possible, and there's some emerging evidence that these goals might be helped achieved as well. So what I'll try to do in this talk is illustrate some of the opportunities by talking about the difficulties, because these molecular omics are different from genomics in at least 3 important ways. First, they're not one thing, they're quite diverse, and therefore they have diverse needs for their measurement. Secondly, they're dynamic, which gives both a temporal dimension, which is an opportunity because they change through time, but also means that there are additional difficulties from distinguishing the signal from noise. And the third thing is that they may well be tissue-specific or tissue-specific context-specific effects that we need to be mindful of. So what I'd like to spend is provide 1 or 2 examples for each of these areas to illustrate some of the challenges. And what I'd like to do is to highlight the experience that we've had in trying to develop a proof of concept molecular omics study, the so-called interval study of 150,000 blood donors in England, which has conducted a range of assays in individuals, including sequencing in collaboration with the Wellcome Sanger Institute, to try to test the value of some of these approaches with the view of trying to perhaps inform what studies like UK Biobank and other larger cohorts might choose to do or not choose to do. So I'll choose one or two domains to illustrate, and the first one being the domain of, if you like, plasma proteomics. So proteins have been particularly difficult to measure in large numbers accurately, even though there's great interest in studying them for obvious reasons. They're the key effector molecules in biology and the targets for most drugs. Where most assays have fallen down is either in throughput or being able to have breadth or accuracy. So that combination has been very difficult to achieve. A company in Boulder, Colorado called SomaLogic some years ago came up with an inventive approach to try to achieve this. What you see here is a so-called aptamer, which is a single strand of DNA, about 20 pairs long, 20 bases long, which can be configured in any way that you like. And these aptamers have an unusual property of being able to fold so that they can bind with these pockets or so-called domains of proteins, but also because they're DNA, they can hybridize to other bits of DNA. They can stick to other bits of DNA. So essentially, they provide the opportunity to take an analog piece of information, a protein binding site, and to convert it into a digital read. So that's the dual property. That was the fundamental insight for this assay. And so a few years ago, we decided to cautiously try to decide whether this would potentially be a fit-for-purpose assay for population scale studies. It is, of course, not an agnostic assay. So the panel I showed you, you might wonder why this says 5,000. The previous slide said 3,500. We used in our study about a 4,000 plex, 35 to 4,000 plex assay. Actually, the latest version, which I think is becoming available, is a 5,000 plex of the assay. But it's not an agnostic assay. As you can see, the proteins which have been put on the panel have a bias towards known biology, and particularly proteins with translational relevance and relate to what people in the field might call the secretome, the subset of the proteins that are secreted by cells. So I'd like to show you a few slides which illustrates not only the thinking about how we were trying to approach the problem of trying to work out the difference between signal and noise in this assay, but how this might generalize to some other molecular omics assays. So an obvious first question is, when you run this assay, what's the variability between assays? And we can answer that question by calculating, of course, coefficients of variation. And this provides a reasonably reassuring answer for this assay. The next question, and this harks back to Rory Collins' comments about regression dilution bias and the discussions we had last night with Richard Pito over dinner, is how stable are these measurements within individuals over time? So if you take a few hundred individuals and take paired measurements 2 years apart, you see that there's not a single answer for these 3,500 proteins, but that actually the large majority are somewhere near the values that we know well for, say, blood pressure or LDL cholesterol, about 0.5, 0.6. So clearly the answers differ for different proteins, but most of the proteins are reasonably reproducibly measured. Next question is, do they pass our test of showing associations, at least with the subset of proteins where we know they're to be associated with established phenotypes? And there were several examples that we saw that increased our confidence that what was being measured in this assay system was meaningful. But perhaps the most broadest and compelling evidence of meaning was the so-called genetic validation of the assay. So in several thousand individuals in the interval study for whom we had genome-wide genotypes, we assessed the 3,500 or so protein readouts that we had. So on this horizontal axis, these are the genetic variants ordered by chromosome. On the vertical axis, it's the proteins that are encoded by those same genes. And I should say this is work that's been, the first author is Ben Sun, who's a PhD student in my group, and several others, and this work is now in press. So each of these dots represents a compelling statistical association which exceeds the usual GWAS standards because of the correction for the number of proteins measured. The diagonal red line tells us about those genetic variants within or in the vicinity, close vicinity of a gene that associate with the protein levels encoded by that gene. So essentially, the red diagonal line is providing us with broad, compelling validation that the assay is measuring what it's supposed to be measuring. The vertical blue lines tell us something differently. They're telling us about a single variant or a locus that's associated with many different proteins. And that's interesting because this is telling us about the body's central protein expression machinery. So we know many of these loci already, such as complement factor H, are the hubs of cascades in this case. Or we already know many of these loci are pleiotropic, such as ABO or ApoE. So this is confirmatory knowledge, but perhaps showing us a broader panel of proteins than before. But many of the proteins, which are not in these neat stacks, are revealing new biology. And this new biology, we think, or has the potential to reveal new biology, can yield new insights and substrates. Now, putting biology aside for the moment, if you're just interested in risk prediction, you can take that validation that I showed you and try to make inferences about how this protein panel or subset of the protein panel can improve prediction of a chronic condition. In this case, the recurrent risk of cardiovascular disease. So a group of investigators in San Francisco took 2 studies of about 1,000 patients each and reported that if you add a subset of 9 proteins from a narrower version of the SomaScan that I just showed you, that you get some improvement you might get some improvement in cardiovascular risk prediction. The authors acknowledge that it was a relatively modest improvement, which is true, and it requires further confirmation, but this illustrates the approaches that people are using to these molecular omics in terms of risk prediction. So of course, prediction is a black box. One doesn't necessarily need to understand fully, other than one has a reproducible signal, what it means. If you want to use these types of omics to help understand biology, there is a deeper understanding of some of the complexities that's sometimes required, particularly for the proteins. And so for etiological inference, I'll illustrate a few questions that we have tried to address, and in some cases partially succeeded, and in some cases the answers are still unknown about this particular assay, about what it can tell us. So one question is, are the readouts that we're getting exclusive to one protein? In other words, is there cross-reactivity? So imagine that you have these so-called aptamer here of this shape. It may bind not only with the protein that you think it's meant to binding with, but also to a slightly different version of the same protein, or maybe another protein. Now, we tested this, or actually the company has tested this in relation to a subset of about 1,000 of the proteins that have considerable homology with other proteins on the panel, and only a small subset have any evidence of potential binding. And actually, most of those relate to the situation where you're looking at 2 different versions of the same protein. So that's somewhat reassuring. Next question is actually a problem which affects all protein binding assays, not just this system, and which has actually been hidden in the literature, even for standard ELISA-based assays. And this is the problem where individuals may have different individuals may have differences in a base that's a coding variant that leads to a structural change in their protein. And so actually, due to that inherited difference, the aptamer, in this case, or some other binding agent, may bind to that individual's protein, but not to another individual's protein. So the readout is telling you not about protein abundance, which is what you're interested in, but just epitope binding affinity. Now, there are computational ways that we've used to take out this effect in our study, and it takes it out for most of the findings. And then there are wet lab ways that one can do this. So for example, you can use a different assay binding system that binds to different domains of the same protein, different pockets, and therefore you're unlikely to see this problem again. But ultimately, the way to properly test this is to use non-binding assay methods such as mass spectrometry, which are very difficult to scale and to use on a wide-angle basis, but they're advancing. But this is an illustration of it's difficult. One can make some progress, but making complete progress, there's further progress that's needed. A further question is the readout telling us about the free protein that's circulating in plasma, or is it telling us about groups of proteins that are sticking together, the protein complex? So here is an example where we were investigating one particular protein, proteinase 3, and we know that one of its ligands is alpha-1 antitrypsin. And so the question that we asked, together with our collaborators at SomaLogic, is this aptamer binding to this complex, or is it binding to the free PR3? And as you can see from this gel, that it's actually the complex. Now, this has only been done for a relatively small set of the proteins in this assay system. And in an ideal world with unlimited resources, one would want to do this for all of the systems to really understand the biology. And more fundamentally, and I guess for all of us who are interested in blood-based epidemiology, is how far can blood take us? What do proteins in the blood reflect? Well, we know that there are more than 200 cell types in the body, and that we know there's increasing evidence of cell type specific and context-specific effects. And of course, the reason for that is that proteins, the origins of proteins come from a number of different aspects of biology. And I think it's important that as we take some of these assays into large-scale epidemiology, is that we're not, as epidemiologists, biologically naive in interpreting these associations. And of course, there are some ways that we can borrow information from other studies, such as reference databases, such as the gene tissue expression database, or the epigenome roadmap database, or the blueprint database, and other databases that are being developed. But I think fundamentally, to drill deeply into some etiological questions, some of the large blood-based epidemiology will need to be supplemented with experimental approaches that gives us cell type specific insight for certain types of questions. Now, having said that, here's one illustration where I think we've made some progress. And again, this comes from the same report. So if we consider an enzyme that's produced by the macrophage, so it circulates in blood, so there it's perhaps giving us some useful insights, but studying plasma, we can construct quite a powerful allelic score made up of some 15 or 20 independent variants at the gene that encodes this protein, and some of which lower the levels of the plasma protein, some of which increase it, as you can see here. So this is quite a powerful allelic score. And then we can take this information into separate database, such as the database that Rory Collins showed you this morning called Cardiogram plus C4D, a global genetics consortium. And using a technique called two-sample Mendelian randomization, we can show this association of these variants with this protein with the risk of coronary disease, showing that there's this inverse association, suggesting that higher levels of MMP12, this protein that's involved in elastase, proteolysis, is perhaps causally associated with coronary disease. This is concordant with some separate routes of information about large vessel stroke that was derived in a separate way. But interestingly, for drug development, where at the moment the idea is to inhibit MMP12 to try to improve chronic lung disease. So this would suggest that there could be a major safety signal if one tried to proceed in this fashion. So moving on from proteins to another type of molecular assay system that illustrates the need to control for non-biological variation. And Nancy Pedersen alluded to this a bit in her talk. So moving from proteins to blood cells. So we're all familiar with Coulter counters. A souped-up version of this is made by a Japanese manufacturer called Sysmex, which also has flow cytometry channels, and one can get some 90 or so readouts, perhaps even more, using this, this system on counts and other characteristics of circulating blood cells. So the question here is really about batch effects. So in large-scale epidemiology, of course, with most assays, even high-throughput assays, it will take quite a long time from the beginning to the end of the assay run. So for example, here, consider the 3 years for the assay run, because in the case of these hematology autoanalyzers, they have to be measured on fresh samples, of course. So this is as recruitment is happening, as the blood is coming into the lab. And you can see these discontinuities here. So on the x-axis is time, calendar time, and here as an example is the volume of red blood cells. And you can see that there are these discontinuities that seem to happen every so often. And if you go back and look at the logs, this is when the machines were recalibrated by the technicians. These are calibration artifacts. And they can be smoothed, of course, using simple statistical techniques to produce this kind of corrected output. Another example are these subtle distortions that can happen to, if you like, for want of a better word, operational effects. So again, many large-scale studies have to be pragmatic, using couriered or mailed samples. And so there are some time differences amongst the individuals who are recruited from the blood draw to the actual assay result. And again, one can record that in studies. If one records it, one can correct for it and try to smooth it. I give you those as an example because although they may seem trivial, when you consider the impact that they have on discovery power. So we modeled this in a set of 50,000 individuals using those uncorrected values for red blood cell characteristics. And you can see there's a more than threefold yield in discovery power for GWAS. So one practical finding, say, from this study where we looked at, in this case, just a subset of the 90 parameters, 36 parameters, and work that was led by Will Astle, who's a university lecturer in my department, revealed really quite an extensive genetic atlas of blood cells, and for those who are interested in this area, showing that there's largely distinct genetic determination of these different cell lineages with some relatively modest overlap as a headline result. Perhaps a surprising finding from the same analysis, again using the same technique of two-sample Mendelian randomization. So creating an allelic score for eosinophils, various eosinophil parameters, we're able to show the positive control result, as you would expect, because eosinophils is known to be important in the pathobiology of asthma, and agents such as IL-5 modulators are used in the treatment of asthma, which have their effects partly through eosinophil count. And we're able to show that the same score, surprisingly, is associated with rheumatoid arthritis, suggesting an opening, perhaps a new pathway of etiology for rheumatoid arthritis, and this is now being actively investigated. Very quickly then, about some of the computational challenges that arise from this. So if we were to look at just the metabolon and some of the other metabolomic assays in the interval study, you can see through simple maths how many calculations need to be done. One of the implications of trying to combine genomics and other omics is that high throughput, high performance computing is absolutely essential to be able to make this type of endeavor workable. So my conclusions are the obvious ones. I think there needs to be careful workup of the different assay systems. They're all different, and they relate to different diverse domains of biology. But there's a lot of opportunity here to try to fill in some of the post-GWAS grand challenge of moving from genome to disease outcomes. And there is active efforts now in a subset of studies that are trying to pool some of these assays. I think I agree with MattNelson, and certainly Sydney Brenner articulated it well, the need to cautiously embrace new technologies because that's really one of the key drivers in science. And I'd like to thank the individuals who actually did the work which I've presented and the funders. And finally, perhaps one final word. When the directory of cohorts is updated, perhaps there's a couple of Cambridge-led cohorts that you might wish to consider adding to that I don't think we had an opportunity to share with you, including cohorts in Bangladesh and other low middle income countries. Thank you very much.
So I think we're now throwing this open to discussion, and one of the kind of themes that came through to me as I listened to all of that with respect to phenomics, with respect to genomics and other kind of assays was around this kind of balance of the perfect versus the possible. What's good enough? Can one make it better? But one shouldn't be too pessimistic. We know that quite crude measures in epidemiology can be very valuable in determining associations, but if we make them less crude, they'll be even better. Yeah.
So I had a question. I don't know whether to send the question to you, Rory, or to Cathie, but one of the things in the UK Biobank, you gave these examples where there's new technologies being applied. You have a subset of 100,000 individuals going to be subjected to these studies, and I was just curious at a practical level, for those studies, was 100,000 picked, was that because of budget, or was that because of a balance between that and people's willingness to participate when you went back to try to get more data from individuals?
Partly budget. So with respect to the baseline assessment, once the baseline assessment was running smoothly, we did get additional funding to do, say, eye imaging in the last 100,000. With respect to the web questionnaires, we ask all 250,000 for whom we've got email, would you like to complete it? And then there's choice. With respect to the imaging, where we're doing 100,000, yeah, it's partly down to how many would be willing to come. but it's also with respect to budget. If we had more imaging centers, people would have to travel less distance, they would be more willing to come, and we could probably do more than that. I don't know, Cathie, if you wanted to add to that.
No.
Mike Gaziano is down there.
So my question is for John. I love your protein array that shows the internal validation along the diagonal and the regulatory protein column. So I think that image, visualizing 36 billion different comparisons on one sheet is wonderful. But what I'm intrigued by is not the columns, but the rows. The rows imply that remote variants are influencing the level of variability of the protein in the population. So does that have potential implications for biologically understanding how these proteins interact? You know, proteins interact with each other. Some of it might be noise, but I wonder if you just comment on variants that are far away from the exon influencing the elaboration of the protein.
It's a big question. So these so-called trans associations are exciting. They may tell us about new biology. They may reflect artifacts that we haven't yet understood. So some of the artifacts could relate to the assay's misspecification of aptamers. So I guess one of the overarching points that I perhaps didn't make in the talk that useful to make now is I think as epidemiologists start to test these different assay systems, there needs to be very close partnership with the vendors of the assays. So we found actually that with that protein assay system that the vendor was very open to a lot of toing and froing. And they've actually changed some of the specification of the proteins on their system as a result of the genetic work, which has suggested that a relatively small set, 5 or 10 proteins had been misspecified previously. So the first thing is to work out whether these trans signals are real and whether they can be reproduced. Reproducing these trans PQTLs, these variants, and that jargon just means variants that are distant from the vicinity of the gene that encodes that protein, right, which may tell us about some kind of new cascade or new complex that we haven't known about. If they're real, they're very exciting. But I think there's a lot of work to try to make sure that they are real before telling elaborate stories about them. Having said that, in the manuscript that's coming out, there are several illustrations where it looks like it's real, where it's opened a new line of biology, such as, say, BLIMP1 in relation to inflammatory bowel disease, et cetera. So I agree with your enthusiasm, but there's a lot of hard work to be done.
So this question is for Josh, Cathie, and Zhengming. It was impressive that all of you are using electronic medical record phenotypes, and Josh in particular, that there are algorithms that you've developed to enhance and refine those phenotypes. My question is really about how do you go about validating those across multiple health system electronic medical records, maybe globally? Have you thought about cross-validation even amongst the three of you that had the discussion this morning, and could you envision the development of a sort of a phenotypic algorithm repository that could be used across the cohorts in the room?
Well, those are great questions, Jeff. I think it's a validating across United States is a challenge, and to think about sort of extending that across the world. I suspect that when we think about the performance of things like codes and probably meds as well, you'll find differences in different countries based on the financial pressures and sort of the documentation pressures and recording practices and electronic systems there. You certainly could validate. I'll tell you, in the eMERGE network, typically we take samples of cases and controls randomly, segmenting those and having them reviewed, chart reviews, looking at the medical record to validate. They generally validate pretty well across different sites, but sometimes you see some differences. And actually, one of the examples we saw that I showed for hypothyroidism, there was lower performance at one site due to a need for more NLP there. Fewer people having comprehensive care there. So even within a validated algorithm, sometimes you'll see 4 sites that are all greater than 90% and one site that's 80%.
I would just add, I think there are great opportunities for us to look at that, and that could be one of the major outputs of this meeting, and that we may find that some of the algorithms transfer pretty well. Others it will be challenging because different coding systems are used in different countries during different eras. So, you know, over here you're mainly using ICD-9 and we're using ICD-10 in the UK now. Eventually we'll move forward to ICD-11 and SNOMED, and we have the very challenging coding system called READ, which is, I think, going to be incorporated into SNOMED, used in UK general practice, which has about 300,000 codes in it that includes symptoms and signs as well as diagnostic codes. But I think there will be commonalities, at least in terms of approach and sharing, for example, NLP and text mining schemes, and looking at the common features that we can transfer from one system to another, and looking at the transfer of that learning, I think is a really important challenge for us to take on.
I think I agree entirely what that Josh and Cathie said. I think in Kadori the challenge is actually it's across 10 different sites. So internally we're always looking for consistency or heterogeneity to try to understand, see whether actually the disease association, for example, with the blood pressure, whether it's similar across different regions, as well. So this actually can review quite a lot. I think technically, I think definitely it's doable. We're all using ICD-10. When you look at analysis, one issue we also need to be aware, it's not just disease phenotype, but also the risk exposure, because that can be affected. Genetic, that's fine, but if you want to look at lifestyle factors, there's all sorts of things that will come to play, will affect the validity or the consistency or whatever. For example, if a very high proportion of treatment, medication, so the association observed may differ quite significantly across different populations, so we should always aware. But I think in terms of the approach, I think definitely it's doable. We're very keen to collaborate as well.
I was just going to say quickly to add on that some of the common data models actually do traverse, ICD-9, ICD-10, ICD-10-CM, and have been used internationally to map to SNOMED. I don't know about read codes though, but I think it's an open hypothesis to do deep validation in them, but there has been some work in that space.
Okay, do you want to go ahead?
Yes, I had a question about just how to deal with biorepository management. I think UK Biobank, it seemed like you alluded to the fact that it was you did a few assays that were centrally decided upon at the same time in everybody, versus I'm with the PLCO trial where generally our experience is doing a lot, actually hundreds of small nested case control studies. Once we find out this person got esophageal cancer or whatever, and doing nested case control studies on really 2 or 3% of the whole cohort with different biomarkers or other specific things. So I wonder if anyone has some comments on what's the best way to do biorepository management in terms of studies.
Well, I could kick it off. I think from the perspective of an individual investigator trying to get publications, the nested case control strategy is a great one. From the prospect of creating a resource for many different kinds of researchers to use, then taking the samples out in a random order so you can get rid of any systematic variation as you do the assays over time, and then run those on everybody, produces, I think, a resource that is of huge value for all kinds of researchers to use for all kinds of different studies. But as I try to illustrate, of course, it's costly to do that in the short term. I would say it's very, very cost effective to do in the long term. And again, it was really my point about procrastination. If I was to do one particular thing differently in UK Biobank, it would be to not have pulled the samples out and spent £10 million of the funders' money in doing 30 or so biomarkers like lipids and CRP and HbA1c, not because they're not important, but because it would be much nicer to wait until the kinds of assays that John Danesh was describing could have been done. And we could have had thousands of proteins, thousands of metabolites all done. So that's why I kind of think the value of procrastination, wait until it can be afforded. And hopefully when one does it at scale, as happened with the genotyping, when one does it at scale, that gets the people who have these systems to push the price down. I believe the exome sequencing, which Regeneron are funding along with other companies in UK Biobank, is running around $200 because they're working out how to do it much less expensively because it's their money. And I think that this is where we could, over the long term, really create huge value for biomedical research. But I mean, I'm throw that open to other people who... yes.
Fran Grotstein, Nurses Health Study. I mean, I just want to kind of reiterate what you said. I mean, for purely financial reasons, our group has always done nested case control studies. But, I mean, if any group could find the funds, you know, to massively do all kinds of sequencing across, you know, incredibly large samples in all of these cohorts would produce a resource, you know, that was orders of magnitude greater than the cost of it. With the only, you know, sort of additional part, I mean, Matt pointed out, you know, the sequencing costs, but in fact, that's just one piece of a whole spectrum of activities that have to happen before the samples arrive in the lab. It does cost the cohorts money to retrieve them, organize them, ship them, and obviously once the data's been generated, it takes, you know, a lot of effort to turn those data into a usable resource. So the costs are much greater than just the assays. But still, you know, if it was possible to create a massive resource of, you know, all kinds of omics data across all kinds of these cohorts here, I mean, the value would just be unprecedented.
And there's also the cost of the data storage, which many people don't even think about when it comes to sequencing. Jeremy.
I think it's a really important point here for people that may be wishing to fund these sorts of studies in the future. And I think one of the most important lessons to take home from biobank, two important lessons. One is the funders have got to be really patient and appreciate that the benefits of these cohorts are going to be felt over the long term, and to not push the individuals and the teams running it or those collaborating with it to get quick wins in the early years in order to justify that funding. So there is a huge amount of work to be done from the funding side to make sure that you're planning this for the long term. And then on the positive side, appreciate that by funding the infrastructure, you will actually leverage your own funding multiple times over many, many years, because others will then come on and fund some of the work, which means you don't have to fund it. So there is a win-win for funders, but you've got to be aware at the start that the benefits are going to accrue over the long term, and not force the investigators to chase publications in their early years. And certainly that would be one of the most important lessons we've learned at Wellcome. I don't want to speak for the MRC, but in terms of UK Biobank.
Yeah, I think that we've begun to see a little bit of insight into how we begin to categorize the value of information here. So I was pleased to hear you mention the storage costs, which you know, is an ongoing and recurring cost. The analytic costs, of course, there is the scientific analysis, which is discovery, which we think is all valuable, but I think there's also going to be ongoing, what you might consider to be technical analytics, which is to improve the quality of the existing sequence. I think we're hearing less of the complaints about, well, we don't want to do genome sequencing now because it's going to be better in 2 years, 5 years, 10 years. That's still probably an issue, but perhaps not as acute an issue. But one of the things that a large consortium like this could do is really begin to look at all the different components of the value of this information across, as we saw, the genotype, exome, genome, and begin to rationally say, well, let's look at this as an investment as opposed to some type of a fixed cost, or that we have to get to a certain cost level and then it's somehow magically affordable. Although the delta in cost, if it's really $200 to do a genome and it's $50 to do a genotype, then I think you can really make a strong argument that now we're approaching something where perhaps we don't have to expend as much mental energy about deciding which one of those things to do. But we need to understand what are all the different components and what are the opportunities to really realize realize the product of the investment over a reasonable time course.
Just add a quick footnote that it's not only the omics analytics cost, but it's significant investment in the phenomics analytics, artificial intelligence, creating data cubes, creating other ways that borrowing from climate change and nuclear physics approaches to analyzing very big and very, very complex and very messy data is not a trivial undertaking either.
Can you hear me? Yeah. Arthur Holden, Genomics Resources Consortium. I think the pace of which you bring new technologies in, like when you sequence, the role of procrastination, I think, as we've been putting together the Genomics Resources Consortium, one of the points that's become quite clear is the ability to use increasingly well-imputed genotyping data sets and integrate that into more of the phenotyping data allows us to think critically about how do we define phenotypes as we move forward. So those periods of procrastination may actually turn out to be very productive periods.
I just want to kind of add the point that it's not genotyping or sequencing, but it could be both. Because the timetable might be different. You know, if you do the genotyping now and you delay the sequencing by a year, probably cheaper than doing the sequencing now, for example. So, and you learn a lot by getting the tractable data early. So I would try to be a little bit cautious about the versus or the or.
Yeah, I think you need the full genome sequences, as has been mentioned before, for those reference panels anyway, in order to do the high quality imputation. So I think both are going to be needed to some extent. Are there any further questions? We've got 1 minute left. So I just also want to, if there are no further questions, just a comment on, so the three of you were talking about EHRs, but phenotype data, so for some of the cohorts, phenotype data is collected as EHRs, but for others, you have that separation between the research cohort, which then collects via CRFs, and there I think it's much harder to harmonize the phenotypes because people are not having standard questions for asking, about the same thing like smoking. You can ask that question many different ways, and it's not comparable. So I think it depends on what cohorts you have, whether it's sort of more research-based or based on the EHRs. So I don't know if you have any comments on that, whether you've come across that issue.
Yes, I think for us, where we've seen that the most would be all of us, in that we have survey questionnaires that we purposely tried to borrow from, UK Biobank, MVP, other projects, in order to keep as much harmony as we could with existing resources, and then we're actually trying to map those to EHR data elements as well, and so they'll be combined through one data table as much as we can. Sometimes there's going to be parent-child relationships and things like that, but it would be nice. You do see lots of sibling concepts related from surveys.
Well, and I just wonder, Josh, if it'd be possible to, nothing is less standardized than electronic medical records, right? So why not apply NLP to questionnaires? There are probably 80 different questionnaires represented in this room, and why couldn't we do that? At least it would be a smaller universe of terms.
Great idea.
Great. Well, thanks very much, everybody.